Section will continue our discussion of cellular adaptation to stress and injury by talking about cell death. When an injury becomes irreversible, this leads inevitably to cell death. And an injury becomes irreversible with the onset of membrane disruption. Once membranes get disrupted, the cell is destined to die. What happens, one way that we can measure the degree of membrane disruption is by measuring the release of cytoplasmic enzymes into the blood. For example, when you have myocardial damage and a myocardial infarction, for example, you can measure creatine kinase or troponin I or lactate dehydrogenase or aspartate aminotransferase, which get released from those dying cells. If you have liver damage, dying liver cells, hepatocytes, release alanine aminotransferase or aspartate aminotransferase, gamma-glutamyl transferase, or alkaline phosphatase into the serum. And so we can detect, measuring these enzymes, we can detect this membrane disruption and the degree of cell death going on. Now here's a schematic of what this looks like. On the left we have a normal cell, and on that cell undergoes some injury, which initially is reversible. You get some damage to the DNA causing clumping of chromatin. You get swelling of the ER and mitochondria. And if the injury at that time is withdrawn, if the injurious stimulus is withdrawn, then the cell can still recover and go back to its normal state again. However, if the injury is persistent or severe enough, then eventually you'll get membrane destruction or disruption, and the cell injury will become irreversible, and the cell will die. Now here's an example of what that looks like. In the previous section we showed this normal renal tubular epithelium and what it looked like when there was reversible injury. Compare that to what you see in this irreversible injury. Here the main thing you see is membrane disruption. There's been disruption of the cellular membrane, and that means that the cell is going to die. There's some amorphous densities, which are clumped proteins, and organelles, which are completely gone. There are mitochondria, which have blown up. The nucleus, notice, is very pycnotic. It's very dense, and the chromatin is very clumped. And that's a result of irreversible damage to the nucleus. Now, there's basically two types of cell death, necrosis and apoptosis. Necrosis is tissue death that occurs as the result of lethal or irreversible cell injury with inflammation. What happens in this case is the cell expands, it's destroyed in an uncontrolled fashion, and that stimulates an inflammatory response. That's necrosis. And necrosis often leads to additional cell damage in the surrounding tissues as well. Apoptosis, on the other hand, is programmed or controlled death of single cells by activation of particular enzyme pathways. And it has no inflammation, and the cellular remnants, rather than getting scattered, are phagocytosed in a very organized fashion. The cell shrinks, the nucleus becomes pycnotic, the cell blebs off pieces of membrane which get phagocytosed, and the cell is eventually consumed. So if you think about it, you can think about it in one way. Necrosis, in a sense, is murder. It's the cell being killed by some external stimulus that it can't repair itself from. Whereas apoptosis is more like suicide. The cell kills itself in a controlled fashion from the inside out, in an organized way. With necrosis, you have inflammation. With apoptosis, there is no inflammation. So here's a schematic of what that looks like. So with necrosis, you get enzymatic digestion, you get leakage of cellular contents because of an external stimulus. With apoptosis, you get controlled blebbing of membrane contents off, which get phagocytosed by macrophages, and there's no resulting inflammation. So we're going to talk about each of these processes in turn, both necrosis and apoptosis. Now, necrosis comes in many different forms, depending upon the tissue and the cause of the necrosis. We're going to talk in detail about both necrosis and apoptosis, and the processes which cause them. Now, there are different types of necrosis, and the different type of necrosis that occurs in any given tissue depends on, number one, the type of tissue it is, and number two, the type of physical injury that's causing the necrosis. The first type of necrosis that we'll talk about is called coagulative necrosis. And this is the type of necrosis that occurs with infarction or ischemia or hypoxia of tissues. And this occurs in all tissues except for the brain, which has a different kind of necrosis we'll talk about later. This type of necrosis the hallmark of this type of necrosis is denaturation or coagulation of proteins in the cell by acidosis. And that's where it gets its name, coagulative necrosis. The macroscopic morphology that you see in this case is one of two things. Either it's a pale infarct, where you have the tissue is dense and pale because of loss of blood circulation to that tissue, or you have a red infarct where you had loss of circulation followed by reperfusion, where the blood has rushed back into a damaged tissue. Microscopically, what you see in coagulative necrosis is an intact cell outline. You can still see the cell borders, but the nucleus is gone. And you can see that in the following pictures. So here's a heart which has been affected by myocardial infarction, and you have necrosis of this tissue. So you see this pale tissue in the myocardial wall. That's coagulative necrosis. Now, another type of necrosis is liquefactive necrosis. This is the kind of necrosis that you get either in an ischemic brain infarction, where you have hypoxia of brain tissue, as shown at the right, or by bacterial infection with inflammation of any organ. So the reason you get liquefactive necrosis is you get digestion of the tissue by hydrolytic enzymes, and that causes the tissue to become liquefied, and then the liquefied tissue is consumed by macrophages, leaving a cavity, a cystic cavity, behind. Now, these enzymes which digest the tissue are produced by neutrophils and macrophages in bacterial infections, for example, an abscess, or by neuroglial cells in the brain, in the case of central nervous system hypoxia or infarction. So here's the difference, a microscopic picture of coagulative versus liquefactive necrosis. On the left, you can see a kidney infarction. This is coagulative necrosis. You can still see cellular structures. You can still see the outlines of cells. But notice that none of these cells have nuclei. And so that's the hallmark of coagulative necrosis. On the other hand, this kidney abscess, you can no longer see any cell borders. You can no longer see any structure. The tissue has been completely digested. And that's the hallmark of liquefactive necrosis. A third type of necrosis is what we call caseous necrosis. Caseous necrosis is caused by a particular type of inflammation that we refer to as granulomatous inflammation. And granulomatous inflammation, at least the kind that causes caseous necrosis, is most typically caused by mycobacterial and fungal infections. And it causes a distinct form of coagulative necrosis. What you see macroscopically in this is a granular, cheesy consistency material, as seen in the picture on the right. A fourth type of necrosis is fat necrosis. Fat necrosis is caused by any release of lipases, which hydrolyzes triglycerides in adipocytes. And you tend to get this in things like acute pancreatitis, where you have you have release of pancreatic lipases, which digest surrounding adipose tissue. Or you can get it in trauma, for example, where you have injury which releases lipases and causes the adipose tissue to be digested by them. Now, what happens is when these triglycerides are hydrolyzed, is you get free fatty acids, and free fatty acids are kind of like soaps, and they tend to engage in a saponification reaction with calcium. So, in addition to the necrosis that you see, you'll also see calcification in these tissues. And so you see these chalky, whitish deposits that you can see in the pancreas here. That's a hallmark of fat necrosis. Microscopically, what you see is the pale ghosts of adipocytes that are filled with this calcified material. A couple other kinds of necrosis. Gangrenous necrosis is when you get vascular occlusion to lower extremities or to the bowel. This is either wet or dry gangrene. The wet gangrene is anytime you have gangrenous necrosis, which is complicated by infection. And so you get liquefaction and inflammation that causes it to be wet and pussy. And that is wet gangrenous necrosis. On the other hand, dry gangrenous necrosis produces primarily a coagulative type change. So you get destruction of tissue without the resulting liquefaction and inflammation, at least not to the extent that you see in wet gangrene. The last type of necrosis that we'll talk about is fibrinoid necrosis. Fibrinoid necrosis is a type of necrosis that's unique to blood vessels. In this type of necrosis, you get it from things like immune vasculitis or malignant hypertension. What happens is you get inflammation, or hemodynamic stress causes damage to the small blood vessels, causes accumulation of proteinaceous material in the vessel wall. And so you get the formation of hyaline-type, hyalinized vessels, as you see in the picture to the right, with this amorphous pink material thickening the vessel wall. And that's called fibrinoid necrosis, and again occurs from immune vasculitides and malignant hypertension. So those are all different categories of necrosis. How about apoptosis? How does apoptosis occur? Now, we already described for you what apoptosis was. It's the genetically controlled, enzyme-mediated, energy-dependent death of individual cells. Remember, it occurs in a controlled fashion. The cell degrades its own nuclear and cytoplasmic proteins and then blebs those things off, and they're all phagocytosed. Remember, there is no associated host reaction, no associated inflammation with apoptosis like you see in necrosis. Now, apoptosis can be both physiologic or pathologic. Physiologic in the sense that the normal turnover of cells in living tissues is an apoptotic process. For example, in your gut, where the cells turn over, that's apoptosis. Your blood cells that turn over on a regular basis, that's apoptosis. Apoptosis also is involved in the programmed destruction of cells during embryogenesis. For example, at some point in embryogenesis, we all have webs between our fingers and toes. But those webs are broken down as we develop, and that occurs by a process of apoptosis. Another physiologic apoptosis is the apoptosis that occurs with loss of hormone-sensitive tissues when hormones are withdrawn. For example, in the normal menstrual cycle, as you get a withdrawal of estrogen and progesterone and so forth, you get a breakdown of the endometrium, and that's an apoptotic process that leads to the normal bleeding in menstruation. Now, apoptosis can be pathologic as well. For example, a virus-infected cell may be induced to die by cytotoxic T cells. DNA-damaged cells also can go through a process of apoptosis. This is normal apoptosis, but it's pathologic in the sense that it's caused by some disease or injury. Now, how does apoptosis occur? Well, cell death is initiated by some death signal. That death signal can come from outside the cell via some cell surface receptor, like Fas and Fas ligand, for example, or from inside the cell via some signal from mitochondria. So the cell either receives a signal from outside that says it's time for you to die, or there's a signal from the inside that says something's not working properly and we need to die. So that death signal leads to the activation of a special class of proteases called caspases. And these caspases initiate and propagate proteolytic cascades which lead to the breakdown of the cell. Now, the caspases activate nucleases, and these cause nuclear fragmentation, breakdown of DNA, and condensation of DNA. And proteases, which degrade the cytoskeleton, cause cells to shrink and break down all the proteins of the cells. The result of that is DNA breakdown and cytoplasmic budding, and this forms those apoptotic bodies which then can be phagocytosed by macrophages. So here's a schematic of how that process might occur. You have the external or intrinsic pathways from receptor-ligand interactions, things like Fas and Fas ligand, a tumor necrosis factor with its receptor. You get the withdrawal of growth hormones and factors, and that causes this external signal. Or you may have an internal signal from mitochondria, like we talked about. Now, these pathways converge upon caspases, and these caspases then initiate this propagation cascade, leading eventually to endonuclease and protease activation and breakdown of cellular contents. Here's a scanning EM image of an apoptotic cell, and you can see in this image the formation of these apoptotic bodies with the cytoplasm budding off, and eventually each of these little apoptotic bodies will come off and be consumed by a macrophage. Now, what happens to DNA in apoptosis versus necrosis is very characteristic then. In both cases, DNA gets broken down. But as you can see in lane B, this is an electrophoretic gel with a stain for DNA. You can see in lane B that there's laddering of the DNA. In other words, there's a regular, stepwise breakdown of DNA by endonucleases. Whereas in C, which is an example of necrosis, there's a much more haphazard breakdown of DNA, leading to just a smear of the DNA as it's broken down into smaller fragments. So that ends the section on necrosis and on apoptosis. Last of all, in this general pathology section, we want to talk about pathologic accumulations in cells. Just a descriptive, this is part of the pathologic process that occurs in some diseases. And I want you to be familiar with what some of these accumulations are and why they occur, so that when you recognize them morphologically, it gives you a clue about the pathologic process that's occurring. Now, one thing that it can accumulate in pathologic processes is what we call lipofuscin. And lipofuscin consists of polymers, of lipids, and phospholipids. And it's a sign of free radical injury and lipid peroxidation. When there's injury to lipids, they tend to form polymers that can be seen as lipofuscin pigment in cells. We also refer to this as wear and tear pigment. This is a pigment that occurs with aging and with wear and tear on tissues and free radical damage that accumulates over time. In certain organs, as they age, you get a combination of lipofuscin with atrophy, and we call that brown atrophy. And again, that signifies wear and tear and free radical injury. Another accumulation that you can get in cells is hemosiderin. This is a pigment consisting of iron bound to the protein ferritin. It's a storage form of iron, and there's two situations in which you can get accumulation of hemosiderin. In both cases, it's caused by iron overload. Now, first of all, you can get it in a systemic overload in macrophages with no tissue damage, and this is called hemosiderosis. And you may see this in normal processes where you have excess bleeding or things like that where you get iron accumulations. There's a more pathologic form of this called hemochromatosis, and this is where you get extensive overload causing dysfunction of tissues and scarring, and that's hemochromatosis. We'll discuss this particular disease in more detail in the liver section of the pathology lectures. Here's a picture of what hemosiderin looks like. In H&E stain, you see a yellow or brownish granular pigment. If you do a Prussian blue stain, which highlights iron in blue, you can see accumulation of this granular blue, bright blue material, which represents hemosiderin. Another accumulation that you can get in cells is calcification. Pathologic calcification is an abnormal deposition of calcium salts in tissues. Now, contrast that with physiologic calcification, which is what you see in bones typically. Now, pathologic calcification can be either dystrophic calcification or can be metastatic calcification. In dystrophic calcification, you get a deposition of calcium phosphate crystals in non-viable or dying tissues. And this occurs even when the serum calcium is normal, because the cells are dying and they accumulate these crystals. Metastatic calcification, on the other hand, reflects a real disturbance in calcium metabolism. In cases like that, you often have hypercalcemia, and because of the hypercalcemia, you're getting abnormal deposition of this in cells. And you see that in things like parathyroid adenomas, which overproduce parathyroid hormone, or paraneoplastic secretion of parathyroid-like related peptide. You can see it in renal failure, or in destruction of bone, or in vitamin D disorders. Any situation in which you get hypercalcemia and disturbance of calcium metabolism, you can get metastatic calcification. So this ends the general pathology lecture on cell death and cellular